All right, so my name is Jerome, and this week I'll be presenting the global chemical impact of the microbiome includes novel bile acid conjugates. This paper is by Robert A. Quinn and Associates. It was published, I forget where it was published, to be honest. Laser pointer. Sorry about that. Laser pointer. There we go. All right, so the purpose of this study was to understand the impact of the microbiome on the body chemistry with more than 40,000 operational taxonomic units as of 2012, and likely much higher nowadays. There is really a lack of understanding of what the microbiome's impact is on the chemistry of the body, and it is known to impact the body. It's just unexplored, and the authors were aiming to address this, to explore this topic via the bile acid chemistries in the body, particularly in the gut, and how this could affect the chemistries of multiple organ systems throughout the body and reflect changes in human disease state. So a little background. Something I pulled from some of my references. This graphic in particular from the second reference just stands in as a starting point to understand the production of the bile acids in the gut. And it, in fact, doesn't start in the gut. It actually starts in the liver. So the liver is actually where the majority of the bile acids are produced, and it's pumped through the gallbladder into the duodenum, which is basically the start of the small intestines, and then throughout the small intestines, you progress into the jejunum and the ileum until you get to the cecum, which is the large intestine, and all the way through the rest. So in this microbiome of the gut, it's understood that there's a wide array of small molecules that are produced and tend to influence the chemistries of the body, and understanding the impact of those chemistry changes on our genes, on other organ systems in our body. A lot of these small molecules make their way into our blood. So it's really important to understand why it's important to study the microbiome in the gut particularly. And particularly a good representation of this is the bile acids that we produce in our liver that make our way into the gut and interact with our digestive system, particularly through mediating or helping the digestion of fats and other metabolites throughout the process and removing waste from our system. So we'll progress right into it pretty quickly with the models. So in this paper, they started the initial study with a group of 8 mice, 4 each of germ-free mice and specific pathogen-free mice. They used the germ-free mice as a control organism compared to the specific pathogen-free, with the idea being that germ-free is supposed to be completely microbiome depleted. But as we'll see later in the study, they're not actually fully microbiome depleted. It's just mostly depleted compared to a specific pathogen-free, which has been treated down to still have a microbiome, just far more reined in by comparison. But additionally, moving on from that animal model that they used, they ended up going into the Global Natural Products Social Molecular Networking database, the GNPS database. And then their primary methods they end up using throughout the paper are the LC-MS and MS-MS mass spec analysis, as well as 16S rRNA gene sequencing. They don't use that as much in the data sets so much as they use it to make some correlations. A lot of that data is represented in the extended figures within the paper. They use mass spectrometry search tool, the MAST tool, to do a lot of their deeper analysis later in the paper, as we'll discuss. And they use cultured human gut microbiomes, microbes, with an emphasis on Clostridia, which we'll discuss later as well. But particularly just these 20 different human gut microbes in fecal cultures, essentially. Skewed towards the Clostridia, as I mentioned, because they'll find later in the paper, or in the paper, they find that the correlation between the bile acids, novel bile acids they end up identifying are strongly correlated with sequencing data representing that the Clish data may be one of the primary sources of it, which they do in fact find to be true. All right, so metabolomics of the model. So what they ended up doing with the mouse model in particular here, as an example, is they took 96 samples across 29 different organs for each of the germ-free and SPF mice. So that's 4 each for a total of 8. And they put it on the 3D maps, various 3D maps to visualize it, including this figure here. This is figure 1A, which shows how they mapped the samples from all over the body and all the different organ types. So it's everywhere from the mouth to the trachea down in through the lungs, the stomach, liver, the jejunum, the duodenum, the cecum, all that through the intestines, through the colon. And then it's kind of mirrored on this side for a separate. It was not entirely clear which side was supposed to be the germ-free versus the specific pathogen-free. To me, it does seem there's more signatures on this side, so I'm assuming that that is the specific pathogen-free model compared to this one over here, which seems to have less signals. And that is, this map was created using data points generated from the figure over here, which is actually extended figure 1A and B, which has the has a PCOA, sorry, modeling system that's principal coordinate analysis that they use to basically 3D plot the variation of different data points they gathered from the metabolomics and the microbiome using their spec data for the metabolomics and their 16S data from the sequencing. And they were able to show that effectively, while you do have some differences, a lot of it overlaps in the metabolomics, so a lot of the same metabolites are being produced, small molecules are being produced, but you do see there tends to be a skew with more SPF molecules being present if you look closely, and particularly over here in this high coordinate, which represents a coordinate moving further away from the PC1, which is your first point that you're using as your foundation for variance. So you have your 3 different points of variation, and these are mapped in 3D compared to those 3 different points, with PC1 being the primary point and representing the largest point of variation. And the further away it gets from that point, the more diverse it is. And similarly down here in the microbiome, this is really a good example showing why they use the germ-free model as a control for this experiment. Is if you look at the microbiome, while the germ-free is not microbiome free, it has a very limited microbiome, and there's a lack of diversity shown here where they're predominantly grouped or skewed towards one side, towards one of the axes of variance. Whereas the data set, while there's SPF data there as well, it really skews off towards one of the other PC3 or PC2 points. So the other points of variation, showing that there's just a greater range of diversity there. So continuing on the metabolomics, they used algorithms to identify the various different unique spectra that they could find from analysis of these various samples, 96 different samples, 29 different organs, all of that from the multiple different organisms. And they were able to identify some nearly 8,000 spectra, with 14% approximately being from the specific pathogen-free mice and 10% from the germ-free mice. So there was a greater variation of unique spectra coming out of those specific pathogen-free mice, which you can represent in the figure B here, 1B. You can kind of see the kind of fuchsia color is the shared spectra, and then across the different tissue types here, down here on the axes, you can see the various tissue types ranging all the way from the skin and ears all the way down to the various organ systems. And you can see the different unique spectra, with green representing the SPF only or SPF unique spectra, and the blue representing the germ-free. And if you look closely, I know the contrast isn't the best, but you can tell that there's definitely more of a skew towards the SPF if it's not predominant. While it's predominantly dominated by the shared spectras, and then down here they break it down as well by the various organs. Representing the number, I believe, of spectra they received from each organ. So you can see there's really a rich number of spectra coming out of the duodenum, the jejunum, and the ileum. So from the GI tract, that's where most of the spectra are coming into play. And there's really large differences noticed, particularly in the lower GI tract. And that's something you see in this figure right here, which actually one that I think is the most important on here, or the easiest to understand. I don't know if I'd say the most important, but it's definitely the easiest to understand, is you have this Shannon diversity index. So basically the higher, more diverse something is and evenly diverse it is, so the more consistent and has this diverse element to it, the higher the score it is. And they basically went through and ranked the system as it goes through the body, starting from the esophagus, through the GI tract, all the way until it exits through stool. And you can see, looking at these spectra, how they basically mirror each other for the first half, or more than half. And it was about the time that they hit the cecum, which is the lower intestine, where it really just skyrockets in the SPF. And the diversity is substantially higher, and more noticeably so in the SPF mice. They also noticed that there was a higher metabolome diversity corresponding to a lower microbial diversity. So this seemingly indicating that there may have been less microbial diversity compared to the metabolomes, but were responsible for a greater portion of metabolomes. So just because diversity of microbial colonization is present doesn't mean that there's necessarily going to be an increase in metabolomes or different small molecules being produced. It'll be kind of the inverse. There's more metabolomes being produced by a smaller set of microbes is what they're seeing. So in continuing with the metabolomics of the model, so the SPF mice experienced a weight loss due to water loss in their GI tract, and particularly also, H2 ACO groups, methyl groups in the lower GI tract. With a lot of this water loss, though, it was associated, or H2 loss, it was associated with bile acids through a known microbial transformation. And the GF mice experience mass loss associated with saccharides in all areas of the GI tract. That's also a known mechanism for bile acid transformations following the deconjugation of the cholic acids. And part of this unique mass gain that inversely was observed in the SPF mice, it was represented about 18.2% of the spectra from known molecules related to glycolic acid. So these glycolic acid derivatives seem to be representing a fairly significant portion of these mass gains, resulting in unknown spectra. And what they were able to elucidate through that, which I'll discuss more in the next slide, was that they were able to explain these mass gains, particularly the C4H8 that they were observing being gained by the molecules by looking at this as the glycolic acid as the base form of it, as GCA. And then in figures 2, 3, and 4, these are the bile acids, novel bile acids that they ended up finding. Like I said, I'll go into more detail in the next slide about this, but they found these 3 bile acids were uniquely new amide bond conjugates of the glycolic acid once the glycine and taurine had been removed, which is a known mechanism that typically happens with GCA as the taurine. There's usually a conjugation of taurine or glycine here, and it ends up getting removed. That's part of the normal process for bile acid chemistries. That's been well studied, relatively well studied and understood for a long time, but they found that there's these reconjugations with different amino acid groups, phenylalanine, tyrosine, and leucine being added into the later portion of this molecule through an amide bond. And that represents an anabolic reaction, whereas typically the deconjugation is considered a catabolic reaction. So that's a new mechanism that was not previously known before. All right, so now I'm going to go into a little more detail. So as I mentioned, glycine and taurine are typically conjugated to the bile acid, and this is normal in both mouse models and humans, and it's known conjugates that occur as one of the four main conjugation or main mechanisms that bile acid chemistries interact within the body and with our microbiome. These new bioconjugates, though, were only observed in the specific pathogen-free mice. Specifically, the phenylalanine, tyrosine, and leucine bonds were identified in place of the glycine and tyrosine, or taurine, sorry, not tyrosine. And these were identified via mass spec analysis and confirmed using synthetic standards that they were able to match up the spectra. That's also located somewhere in the extended data for the paper. If anyone was curious to look at that, that starts to get into that range. It's really deep diving into the nerdy stuff. But these bile acids were found uniquely in the duodenum, the jejunum, and the ileum of specific pathogen-free mice, while as the glycine in those areas only. And while you would find glycine and taurine in the gallbladder, the liver, and all of the GI tract. So this kind of represented that these specific compounds were only appearing within this part of the upper GI tract, particularly in the duodenum post gallbladder. So once they were found lower than the gallbladder was introducing the bile acids. So that was indicating that the modification was occurring within the small intestines and not further up in the gallbladder or potentially in the liver itself, where other mechanisms of bile acid chemistry interactions are known to happen. So this suggests that it's not necessarily the host's own mechanisms that are causing these reactions and creating these new novel bile acids, so much as some of the microbes. So I'm going to call them by their short name right now, just to keep this kind of quick and easy. But the fecal was found to be the most abundant in all of the conjugates, dropping off at the cecum. So it effectively disappears at the cecum. And in fact, down here in this figure that I pulled from 2B in the primary research, you see how going down the GI system, most of these novel conjugates are introduced after the gallbladder, which is supporting that these acids do not exist anywhere else in the body and are being produced or modified by the microbiome, and they persist through the duodenum, the jejunum, and the ileum, and then they seemingly dissipate or disappear by the time they hit the cecum. So that leads to the question of these must be interacting in some way or either being metabolized by the body or being reabsorbed into the body through the peripheral organs or blood system. And the author used some of the fecal matter cultures at this point, as I mentioned earlier, as one of their methods. They were able to see that there was no conjugation occurring within these cultures of these particular compounds, the fecol, the tiercol, and the leucol. These cholic acids were not being deconjugated, which while the GCAs, the glycolic acid, was being deconjugated in a known mechanism mediated by enzymes present within the human microbial system. So this represents a new compound that doesn't quite interact in the same ways with the existing microbes in the gut. Yeah. And this kind of represented some new amide linked conjugate identified that breaks 170 years of bile acid chemistry standard. So we, nearly 170 years of history of understanding the bile acid chemistries and how they interact, and particularly that there are enzymes in our body or specific interactions in our body that modify the chemistries of the bile acid consistently, and these are new or novel bile acids that had never been seen before. So it kind of breaks the standard of what we knew to be the limits of bile acid. So how's this translate to humans? Well, the author went on and did a deep dive analysis using some of the databases I mentioned earlier, the GNPS, and they were able to use the MASS tool to search over 1,000 publications in the GNPS, and they were able to find 28 studies with spectra that matched the new conjugate bile acids from both human and mice samples. And just to give you an idea, it was as high as 25.3% of human samples were detected within those 28 studies, showed some signature of spectra for these chemicals or for these new compounds, and nearly 60% of mouse samples had matching spectra. Those are the high ends. The average numbers were much lower, but in some studies they were relatively abundant, just to give you an idea of that. And there was a higher frequency, particularly noticed in these data sets where they were detected in patients that had IBD and cystic fibrosis and in infants. So they pulled data sets from the American Gut Project, which is represented down here as the first chart here and from figure 3A. And that you can see basically the percentage of samples from the sample set here. So for the American Gut Project, it's 490 samples. And you can see there's relatively low levels, but there are detectable spectra representing, a percent or two, or maybe a fraction of a percent for some of these, for the phenolic and tyrosolic acid here, and the glycolic, which the glycolic is, as I said, is a known bile acid conjugate that is known to exist. But more noteworthy is in the American Gut Project, they had 173 IBD patients, 155 cystic fibrosis patients, and then a large number of infants. And you can actually see a relatively surprising percentage of these patients had these known spectra show up within their samples, particularly for IBD, cystic fibrosis. The infant one, they didn't go into as much detail describing why the infants, or trying to attribute why infants had as much of this, compared to these two other patient bases, which are disease states. So I wish they had kind of gone a little more into detail there, other than them likely just being somewhat overlapping or associated with similar GI issues. On the second chart within this graphic, though, this represents data from a cystic fibrosis study where they were able to show that reanalyzing the spectra from that study, they found that a large percentage of the cystic fibrosis patients had spectra matching these new conjugate bile acids, whereas compared to the non-CF patients that were used as control in this study, had effectively none of them other than the glycolic acid, which again, that's a known normal bile acid conjugate. All right, so continuing the translation to humans, the authors looked at another study in particular that was looking at cystic fibrosis, that was the previous one. They looked at another one that focused on IBD and specifically Crohn's disease and ulcerative colitis. And they were able to find that it was specifically people with Crohn's disease that seemed to be more impacted and be producing higher levels of these conjugate bile acids compared to ulcerative colitis or IBD patients. So here you have they ran two different statistical analyses here and plotting the abundance of these compounds. And you can see that the control non-IBD patients here and the UC patients are relatively similar with some more variance in UC. It's a disease state, so you're probably going to see a bit more variance in within the different patient samples based on severity of the disease state. But you notice that the Crohn's disease patients had a noticeable increase that's statistically significant over the basis of patients who didn't have cystic fibrosis or Crohn's disease. So Crohn's disease just seems to be more prone to the production of these compounds. This could indicate a link between the microbiome of the individual with dysbiosis and their novel bile acids, compared whether that be just a simple correlation of the disease state facilitating the growth of these bacteria that may be producing the conjugate bile acids, or whether that's a symptom of it. Maybe it's the colonization of these bacteria producing conjugate bile acids that is promoting the disease state. There's no real way to tell right now, but that is an important question later. So they were able to confirm that this was in fact a microbial product as well, and not just simply produced by the human gut biome, as we kind of saw earlier with the fact that the new conjugate bile acids do not seem to interact with the existing known healthy human microbiome enzymes that are typically present. And in fact, they were able to basically use radio-labeled precursors, force feeding them to mice or through gavage feeding tube, and track the production of the phenylalanine cholate and the tyrosine cholate, specifically by these Clostridium boltiae strain and another strain CC43001B, and that they were able to synthesize those consistently. And they were able to detect the products in the mouse feces, and through analysis confirm that they were in fact the phenylcholate and not just the precursors. So the author does also go through and point out that there's an association between the Clostridium boltiae with potential health outcomes that could be important for further understanding of these interactions. In specific, they mention a link between Clostridium boltiae and autism, as with a large portion of autism, the autism population, particularly children, having this bacteria present within their GI tract, as well as this bacteria being associated with displacing vancomycin resistant E. coli, which also is an interesting health implication there that could potentially be explored later. Oh, sorry. All right. And additionally, they wanted to double check look into what this chemistry change in the bile acids was impacting the body as. So in order to determine this, they used a gene expression assay and potency assay. It used a luciferase reporter assay and HEK293 T cells, a relatively common cell line for potency. And the phenylcholate they were able to find was twice as strong as a naturally occurring and understood farnesoid X receptor FXR agonist and the CDCA. I'm not even going to try to pronounce that one. And it was found to be about twice as strong, while they also found that the tyrosine cholate was the most potent, substantially higher or lower EC value compared to either of these. So absurdly more potent by comparison but keep in mind that the phenylcholate, it was found to be the most abundant. But tyrosine cholate is still relatively abundant comparatively with the phenylcholate. So keep that in mind. They were also, in order to determine the potency and the impact of this on the organism, they used feeding methods like the gavage to feed these compounds to the mice. And they were able to check the expression of the FXR gene and found that these bile acids interacted with the system in a way that changed the expression of FXR effector genes, FGF15 and SHP, as well as the bile acid conjugate, bile acid synthesis genes, with it increasing the effector genes and decreasing the expression of the synthesis genes, which makes sense. These effector genes interact with and repress the synthesis genes. And one unique thing with the increased potency in here and the way it was stimulating these effector genes is that these bile acids are far stronger at repressing the bile acid production in the liver. So, conclusion of this study was that they described a fifth mechanism of metabolism in vivo previously undiscovered and found it to be supported in both mice and human data. And they described the new conjugate bile acid, how the new conjugate bile acids could stimulate the FXR receptors and effector genes, but then they repressed the bile acid production in the liver. And they emphasize the importance of the impact of these chemistry changes in the human microbiome and how they affected the body, and particularly the fact that it modulated the gene expression and production of the bile acid. And how it has implications on the overall health of the organism. So without a real strong understanding of these things, they wanted to emphasize that it's really important for us to elucidate more information about this, because right now, as it stands, we don't understand whether this is an impact of the disease state, or whether this is a symptom of the disease state, or whether it's a causative agent of the disease states. So what information can we draw from this study? The author makes the connection of the presence of these novel bile acids in the human health and how some of these organisms are tied, like I mentioned earlier, to things like autism or displacing antibiotic-resistant bacteria within our gut, both having completely different associations. So understanding the presence of these bile acids in the human health state would be good. And the importance of the microbiome diversity on the chemistry of the body, especially with how it interconnects with our gene regulation. And there could be a link between our microbiome that's not only that could be causative or could be symptomatic of a disease, and how the interplay between the two could affect the outcome of the disease, whether it could be beneficial to increase the diversity of the microbiome or reduce the diversity of the microbiome. And one of the studies points I forgot to mention earlier, they were able to show that depletion of the microbiome removed or lowered the detectable limit of the conjugate bile acids in the mice models to undetectable levels. So clearly depleting the microbiome does affect what biochemistries are being interacted with. And it also shows us that the power of these types of studies and analytical methods and more advanced tools can interconnect data sets from many different studies, and we can really start to discover more links to the human health outcomes. So potential new questions. What is the relative abundance of the microbiome? What does displacing bacteria that produce these conjugate acids have an impact on the host, negative, positive, negative? What kind of techniques, such as fecal matter FMT, could improve the health of the individual with dysbiosis or not? And is there a link between the change in chemistries and neurological disorders? So we know it impacts the body, but how far, how extensive? They were able to show relatively increased signaling changes in gene expression modulation far throughout the body and impacting most organ systems. So we don't really know how far. It'd be interesting to know. And could the changes in this chemistry be a potential new avenue for medical research? All right, and here's my various references. All right,